Bismillahirrahmanirrahim. My name is Kaina Damna. I am starting today Biochemistry. Biochem, first of all, we will study about the chromatin structure. What we will study in chromatin structure. See, there is two types of chromatin. DNA exists in the condensed chromatin form to fit in the nucleus. DNA loops twice around the histone corner of the nucleosomes, beads, and HI binds to the nucleosome and the linker DNA, thereby stabilizing the chromatin fiber. This is a DNA linker. It's a helical like this. Protein is here, H1 histone linker, and it is the stabilizing the chromatin fiber. There is only one simple thing, and this is the DNA loops twice around histone. This is the twice around the histone. See here, DNA has negative charge from phosphate group, and histone has positive charge. Lysine and arginine. In mitosis, DNA condensed to form chromosome. DNA and histone synthesis occur during S phase. DNA and histone synthesis occur in S phase, one line, and here DNA negative charge, arginine and lysine of histone is positive charge. They are binding due to opposite charge. Mitochondria have their own DNA which is circular and does not utilize histone. Mitochondria have their own DNA. Okay, one is nucleus and one is mitochondria, which is circular and does not utilize histone. And mitochondria's DNA is not utilize histone. Three things: positive negative charge, S phase synthesis, and mitochondria have DNA which is a circular and not utilized by histone. Here we see the DNA double helix, more zoom DNA, its DNA double helix structure. This is the linker H1 histone protein. These both two are euchromatin histone acetylation, and this one is nucleosome. We call it a whole as nucleosome. DNA and histone protein combined is called nucleosome. H2A, H2B, H3, H4 multiplied by 2. This is the nucleosome, and here we see this is the heterochromatic. This is the condensed form. This is separated. It's eukaryotic and it's a condensed. It's histone deacetylation and supercoiled structure. This is a supercoiled structure. Then it is goes to metaphase chromosome. Now we will study the heterochromatin. What is euchromatin? What is DNA methylation, histone methylation, histone acetylation, histone deacetylation. First of all, we will study about the heterochromatin. Condensed appear darker on EM, label H in nucleus, sterically inaccessible, thus transcriptionally inactive, increased methylation, decrease acetylation. Okay, what is we understand in this line is condensed appear darker on इलेक्ट्रॉन माइक्रोस्कोप में हमें कंडेंस नज़र आता है, and inaccessible, thus transcriptionally inactive, but it's inactive and increase methylation, decrease acetylation. In heterochromatin, this is heterochromatin, इसमें methylation ज़्यादा है, acetylation कम है. यहाँ पे deacetylation भी है. ये हम आगे पढ़ते हैं acetylation और methylation में. Here we see heterochromatin is equal to highly condensed hidden chromatin and bar bodies inactive X chromosomes. इसमें X chromosomes जो हैं वो inactive होते हैं, may be visible on the periphery of nucleus, may be nucleus की periphery में नज़र आए. What is euchromatin? It's less condensed, appear lighter on electron microscope, labeled in E diagram, and transcriptionally they are active and strictly accessible. They are less condensed, unwinding and transcription इनमें इसीलिए हो सकती है. And we will see U, it's mean truly transcribed. Euchromatin is expressed. Now we will see DNA methylation. What is DNA methylation? What we have to understand in this topic is reversible changes the expression of the DNA segment without changing its sequence involved with aging, carcinogenesis, epigenetics, genomic imprinting, transposable element repression, and X chromosome inactivation and lionization. In DNA methylation, we will see the reversible change of DNA without change its sequence. We will see reversible change, a little bit change. We can say mutation is the permanent change. Like in aging involve हो सकती है carcinogens, epigenetics, genomic imprinting, transposable elements repression और X chromosomes इनएक्टिवेट वगैरह हो जाए तो ये रिवर्स हो सकता है डीएनए का जो रिवर्सिबल चेंजेस आते हैं। इसमें हम देखेंगे डीएनए इज़ मिथाइलेटेड इन इम्प्रिंटिंग इज हिडन जीन and methylation within gene promoters CpG islands typically silences gene transcription, repress कर देता है ट्रांसक्रिप्शन को मिथाइलेशन जो है, और CpG मिथाइलेशन मेक्स DNA को म्यूट कर देता है। डिसरेगुलेटेड DNA मिथाइलेशन इज़ इम्प्लिकेटेड इन फ्रेजाइल X सिंड्रोम में ये इम्प्लिकेटेड होता है। लाइक दिस इट्स अ methylated form. Here it's not like not replicable, it's a mute kind of DNA, इसमें mute हो जाता है। We will see the DNA methylation and then we will see hidden methylation. What is hidden methylation? Usually causes reversible transcriptional suppression but can also cause activation depending on location of methyl group. ये आमतौर पर किसकी वजह बनता है? रिवर्सिबल ट्रांसक्रिप्शनल इसको सप्रेस करता है बट नॉट काज ऑफ एक्टिवेशन डिपेंडिंग ऑन लोकेशन ऑफ मिथाइल ग्रुप। लेकिन मिथाइल ग्रुप की प्रेजेंस इफ़ेक्ट नहीं करती ट्रांसक्रिप्शन को। और यहाँ पे है इसमें वो होते हैं हिस्टोन मिथाइलेशन मोस्टली मेक्स डीएनए को म्यूटेट कर देता है। लाइसिन आर्जिनिन जो है वो रेजिड्यूज़ हैं किसकी? हिस्टोन्स की, कैन बी मिथाइलेटेड, ये मिथाइलेटेड होती हैं।